Hello, and welcome to MCB 102 Part 3. Last time, we went over the different levels of structures that take us from a bunch of amino acids to a fully operational protein. In this video, we will focus on protein purification and identification techniques to analyze a sample that may contain thousands of different proteins. The goal of protein purification is to remove non-protein impurities and isolate a specific protein from a source, whereas identification serves to identify a target protein in a sample and verify that our protein that we've isolated is the one that we wanted. So the first step in protein purification is to choose an appropriate buffer system that matches the environment the protein normally resides in inside the cell. In general, you want to keep the pH around the pH where the protein operates normally, and you will also want to include salts to neutralize charged proteins to avoid aggregation, reducing agents to prevent oxidation forming any disulfide bridges, protease inhibitors to prevent the protein from being broken down and degraded, and detergents if the protein you're interested in is bound in a membrane. The next step is cell lysis, which rips apart cells to release the protein inside of them. This can be done in a variety of ways, including blenders, homogenizers, or even a simple pestle and mortar. Afterwards, the sample undergoes clarification in order to separate different components of the cell. This process is done by centrifugation. Generally, larger objects like organelles will be separated out first, and proteins will be the last to be separated. Once the proteins are isolated, we can now separate the target protein by taking advantage of its unique properties based on its size, charge, ligand binding affinity, and solubility. Some variants of column chromatography are very useful for this. Absorption chromatography, aka ion exchange chromatography, separates positively and negatively charged proteins from each other. The walls of the column are positively charged in an anion absorption column, since it binds to negatively charged proteins. And on the other hand, the walls of the column are negatively charged in a cation absorption column. Another type of column is the affinity column. The column walls here are lined with an immobilized ligand, which only a specific protein can bind to. As the sample passes through the column, the desired protein will be trapped in the column, while the rest of the sample passes through. Finally, there is the side exclusion column. In this column, the column is filled with porous beads. Each bead contains a convoluted network within, which is small enough so that large proteins cannot enter, and so they will go around the beads and emerge from the column first. On the other hand, smaller proteins can fit inside the bead, and so they will take a much longer, windy path going through the beads and emerge from the column last. With these three types of column chromatography, you can use them in a wide variety of combinations to isolate almost any protein you desire. Having successfully isolated your protein, now your goal is to quantify your protein and identify its presence or verify that you have isolated your desired protein. So, if you just want to figure out whether your protein is there in the sample, a colorimetric assay is commonly used. These involve a dye or a transition metal that changes color in the presence of the protein. By making a standard absorption curve for the protein using known quantities of the protein, you can determine the concentration in your sample by comparing absorbance values to those on the standard curve. Another way to determine protein concentration is to use UV absorption, which works because tryptophan, tyrosine, and cysteine all absorb UV light strongly. So, if your protein sample happens to consist of multiple proteins, you can use polyacrylamide gel electrophoresis, also known as PEACE, to separate proteins by using an electric current to drag them through a gel matrix, which slows down larger molecules more so than smaller molecules. Native PEACE leaves the proteins intact, so their quaternary structure remains present, making it useful to check that the protein is all in one piece. Thus, native PEACE separates proteins based on their shape. On the other hand, SDS page uses a detergent called sodium dodecyl sulfate, abbreviated SDS, which denatures a protein and completely unfolds it to form a straight chain. Thus, it allows a protein to be separated solely based on the length of the polypeptide chain and eliminates protein folding as a possible variable. Finally, you can also use 2D page to improve protein separation by taking out one lane from a completed page and then placing that lane at a 90-degree angle in a second page. So the second page is typically a different type than the first page. So for example, if the first page completed was a native page, then the second page would be most likely SDS. Another separation technique we can use is called isoelectric focusing, which separates proteins based on their isoelectric points, which is the pH where the protein has no net charge. Each protein's pI depends on the amino acids that make up its primary structure, and the isoelectric focusing gel is a strip containing Zwitter ions that are strong buffers, which allows the gel to maintain a pH gradient across the gel. So any protein placed on an IEF gel will move along until it reaches the pH along the strip that matches the protein's pI. In situ assays can be used on enzymes to determine the position of a protein on the gel. However, enzymatic activity requires that the protein be folded correctly, so this can only be used on a native page. By adding a substrate that changes color, once the enzyme changes it, it lets you identify the location of the protein on the gel. On the other hand, a Western blot can be used to determine the location of proteins even on an SDS page. How it is done is, the sample is first exposed to a primary antibody, which binds to the target protein. Next, a secondary antibody, which detects the primary antibody, is added to the protein in the SDS page. The secondary antibody has an enzyme attached to it, and applying an assay to the attached enzyme will reveal the location of the protein. Like I said earlier, this works on denatured proteins and in SDS page. The last technique we'll cover is mass spectrometry. This separates proteins by their mass to charge ratio. First, the sample is vaporized and placed into an ionization chamber, where the electrons are knocked off of the sample to make it positively charged. Then, a positively charged repeller pushes the sample and accelerates it into a magnetic field. The magnetic field deflects the path of each particle that passes through it to a degree that depends on the magnitude of its charge. Therefore, molecules with higher mass to charge ratios will be deflected less, and different mass charge ratio samples will hit the detector at different locations. So the detector is able to distinguish between different proteins that are only separated by just one amino acid in overall mass. And that concludes protein identification.